Hello Biology 100 students, and welcome back to our discussion of the cell as the fundamental unit of life. In this lecture we're going to discuss membrane processes. But before we go to that, I want to do an overview and review of the different functions of the cellular organelles that we talked about last time. So what we have here is a cellular model from probably the 1950s over there. It's pretty old, but it's what we have in our department. So let's go through the parts of the cell from outside to inside. First of all, outside the cell here we have our plasma membrane, and the plasma membrane is made up of phospholipids, some proteins, and also cholesterol. And the purpose of the plasma membrane is to regulate passage of things into the cell. It's a selectively permeable membrane, as we're going to talk about today. Now, inside the cell, everything between the plasma membrane and the nucleus is called cytoplasm. And the cytoplasm includes all these different organelles in here, as well as the cellular jelly, which is called cytosol. Now, starting at the very inside of the cell here, we have the nucleus. And remember, the nucleus is a membrane-bound structure we only find in eukaryotes, and it's where we have our DNA, which is the instruction set to make a whole new cell, but it also contains the information to make proteins and things like that. So inside the nucleus, you'll see a darker structure called the nucleolus, and the nucleolus is where we make our ribosomes. Okay, so going outside the nucleus, what do we see here? Well, we have some endoplasmic reticulum. What I can see here, it has little bumps on it, and so these bumps on the endoplasmic reticulum probably mean that it's rough ER, because the bumps in this case, the white bumps, would be ribosomes. So the rough ER, if you remember, manufactures proteins for use outside the cell. Okay, what other structures can we see here? Well, if I go up here, here's an organelle we actually didn't talk about last time. It's called the centrioles. The centrioles help the cell to prepare for division, and they help to divide the chromosomes during the process of mitosis. And we're going to be talking about that in the next lecture or so. Okay, further up here, not quite sure what this is. It could be smooth ER, it could be a Golgi apparatus. If it's a Golgi apparatus, we say that it's used for the FedEx of the cell, used for packaging and shipping. If it's smooth ER, then it would be used for lipid synthesis. Sometimes these older models, it's hard to tell what is what. Okay, so what do we have right here? These structures right here, the red hot dog looking structures, everybody knows what those are. Those are the mitochondria, and those are the powerhouse of the cell. They're used to produce ATP by breaking down things like carbohydrates and proteins and also lipids. So they produce ATP, and they have a double membrane structure there. Now, what else can we see inside there? Well, up top we see what looks like a vesicle that's either entering or exiting the cell. If it's exiting the cell, it would be an exocytotic vesicle or secretory vesicle. If it's entering the cell, it would be an endocytotic vesicle. Again, we've got more mitochondria up there. That looks great, that looks great. Other structures we can see in here, we definitely have some vesicles. So down here, this is a type of vesicle organ. It could be a peroxisome or a lysosome. I don't know, I can't tell. But remember, the lysosome is the cellular stomach. It's responsible for digesting things, breaking down worn out organelles, whereas the peroxisome is responsible for metabolizing certain metabolic waste products like hydrogen peroxide. Now, depending on the different models that you look at, you will be able to see some structures, but not other structures. But suffice it to say, you should be able to identify mitochondria, rough and smooth ER, Golgi apparatus, lysosomes, and also the nucleus and plasma membrane. Okay, so now that we've talked about the different functions of the organelles inside the cell, we're going to talk about how molecules move across the cell membrane into the cell. And it's going to be divided into 4 topics. First, we're going to talk about diffusion, which is a simple process where things move from an area of high concentration to low concentration. We're going to talk about other forms of passive transport, such as osmosis, which is the diffusion of water. We're going to talk about active transport, which utilizes energy. And then last but not least, we'll talk about endocytosis and exocytosis. Okay, so remember what we said before, that the plasma membrane is selectively permeable. It allows certain substances to pass through the cell membrane, but not others. Think of it as being a bouncer at a very trendy nightclub. It might let her in, but it may not let you in. So it's semi-permeable. So molecules move through the membrane in 3 different ways. One, through passive transport, i.e. diffusion. They go from an area where they're highly concentrated to an area where they're not highly concentrated. They can also use something called active transport. That is, we can push things against their concentration gradient from an area of low concentration to an area of high concentration, but that's going to require some ATP or energy. And last but not least, we'll talk about endocytosis, exocytosis, which is used to move very large molecules across the cellular membrane. Okay, first we need to talk about passive transport processes, and the first one of these is diffusion. So diffusion is the natural tendency of a substance to spread out, moving from an area of high concentration to an area of low concentration. So look right here where we have some dye molecules. We're dropping them into a beaker. Initially, they're most concentrated in the center of that beaker, but over time they begin to spread out as the water molecules begin to spread them out due to normal Brownian movement. Eventually, those dye molecules are going to be equally spread out all over the entire container, and this is how diffusion works. It works by dispersing things from an area of high concentration to an area of low concentration until these solute molecules are equally distributed. So before we go on, we need to cover some diffusion vocabulary. So first of all, remember that a selectively permeable membrane is a membrane that allows some things to cross but not others. Think about that bouncer at the trendy nightclub. A solute is something we haven't talked about before. A solute is anything that's dissolved in a solution, and usually the solution here is going to be water. So anything dissolved in water is going to be called a solute, and the water is our solution. All right, a concentration gradient means that we have more of a substance on one side of a room or one side of a membrane than we do on the other side. Okay, so this slide just shows an example of a concentration gradient. Take a look right up there. You can see that we have a membrane in the middle and that we have more stuff on the left side of the membrane and nothing at all on the right. So by simple diffusion, if these solute molecules can cross, they're going to go from an area of high to an area of low concentration. And that's just what happens down here, going from high to low. And eventually that's going to proceed until we get equal concentration of our solute on both sides of the membrane. And in this case, we are now at equilibrium. Okay, now let's look at another example of simple diffusion. What we have here is a U-shaped tube which is filled with water. And into that U-shaped tube we have put sugar molecules. We put 12 on this side and we put 4 on that side. So we have 12 on the right side, 4 on the left. Now what's going to happen via simple diffusion? Well, via simple diffusion, things should move from an area of high concentration to low concentration. So since there's more sugar on this right-hand side, it should move from right to left. And that's exactly what happens. Okay, the net diffusion of sugar spreads out until we have an equal concentration of sugar on both sides. So net diffusion was from the right side of the tube to the left side of the tube until we eventually reach equilibrium. Now we're going to talk about a special case of diffusion called osmosis. Osmosis is the diffusion of water across a semi-permeable membrane. Now water diffuses just like everything else. It goes from an area of high concentration where there's more water to an area of low concentration where there's less water. But now we have to think about solutes because solutes affect which way water is going to go. If we have a side that has more solute, by definition it has less water, and so water will go towards that solute. If that gets confusing, just remember one thing.
Solutes suck.
That is, solutes will always suck water towards it. So if we have an area of greater solute concentration, that's where the water is going to go. Okay, so here's the same scenario we talked about just a few minutes ago. We have our U-shaped tube here, and we have 12 sugar molecules on this right side and 4 sugar molecules on this left side. The only difference now is we've separated this tube with a semi-permeable membrane, and I'm going to tell you this semi-permeable membrane will allow water to cross, but it will not allow our sugar molecules to cross. So the question is, what is going to happen? Well, if the membrane wasn't there, right, our sugar would move from right to left, spreading out, going from an area of high concentration over here to an area of low concentration over there. But remember, I said the sugar can't move because the membrane is not permeable to sugar. So the question is, what's water going to do? So where is water more highly concentrated? Well, water is more highly concentrated over here. It wants to go from an area of high concentration to an area of low concentration. It's more lowly concentrated here because a lot of that room is taken up by solute. Again, if that's too complicated to think about, just remember,
solutes suck.
That is, we have more solutes on the right side and it's going to suck that water towards it. And that's exactly what we see through the process of osmosis. The water has been sucked from the left side of the tube to the right side of the tube because the right side of the tube had more of our sugar molecules, it had more solute, and what do solutes do? Solutes suck. Okay, so now that we know how diffusion works and how osmosis works, let's talk about describing differences in concentration gradients across a membrane. So each of these figures shows what's happening on the inside of the cell and the outside of the cell. Okay, if the concentration of solute is equal on both sides of the cell, we say that those concentrations are isotonic. Iso here meaning the same. So we have the same amount of sugar or salt here, same amount of salt here. And because of that, we have no net movement of water. Now, water can be moving back and forth, but it's not moving any more in one direction than the other. Now let's look at a cell that's placed in a hypertonic solution. Hyper means greater than. So here's the solute concentration inside the cell, and we can see that it's a lot more concentrated outside the cell. Again, we have lots of solute out here, less solute in here. What do solutes do? They suck. And so the solutes are going to suck water out of the cell, causing it to shrink and crenate. Okay, last but not least, we have a hypotonic solution here. Hypo means below, and so this solution has less solute than what's inside the cell. And again, we have more solute inside the cell, and what do solutes do? They suck. It's going to suck water into the cell, and that cell is probably going to swell up and potentially burst. So it's important to remember that water is always moving across cell membranes, even though we might be at an isotonic concentration. When we're at an isotonic concentration of solute inside and outside the membrane, it just means that water is moving in at the same rate that it's moving out. So take a look at this figure here. Water moves in, water moves out. The arrows are the same size because water is moving into and out of the cell at the same rate. So this figure just shows something similar. We have 3 different beakers here, and each of these beakers has a different solute concentration. So let me start with the one on this side. We have a red blood cell, erythrocyte, that has been placed in an isotonic solution. Remember that iso means the same. Because it has the same concentration of solute, the water going in with the arrow is the same as the water going out. So this cell, even though water will move across it, will not change in size. Okay, now let's look at a cell that's placed in a hypotonic solution. Here you can see we have a lot more water going in and very little water going out. And that's because the cell has the majority of solute in the solution, and the cell is sucking the water in, so the cell is going to swell and potentially even burst. Finally, let's look over here on this side. We can see a cell that's placed in a hypertonic solution. Remember that hyper meant greater than. So this is a salty solution outside, and because it has more salt or solute, what will that solute do? Well, that solute will suck, and so water will be sucked out of the cell more so than going into the cell, and so that cell will shrink or crenate. Okay, remember that diffusion and osmosis were both forms of passive transport. They utilize simple diffusion to move molecules across the cell membrane. Now remember that passive transport, like osmosis, like diffusion, does not require any net energy input. We don't have to add energy or ATP into it, but it does require a concentration gradient, meaning the most you can get on one side of the membrane is about 50% of the different solute molecules, because once we reach equilibrium, things essentially stop moving. We have no net movement. So there's 3 different types of passive transport we're going to talk about. The first of these is diffusion through the phospholipid bilayer. The second is diffusion through protein channels, and the third is facilitated diffusion. Okay, first of all, let's talk about diffusion across the phospholipid bilayer. What can move directly in between the phospholipids? Well, generally, whatever it is, it has to be very small. So we're talking about gases like oxygen, carbon dioxide. It can't have a charge, so it can't be any type of ion or anything like that. And it can't attract water towards it. Because remember, the inside of this cell membrane here is made up of all these hydrophobic tails, and they're going to want to exclude water. So primarily, very small nonpolar molecules such as oxygen, carbon dioxide can move very easily across it. The other thing that can move very easily across the plasma membrane are other lipids. Remember, the plasma membrane was formed through phospholipids, and what dissolves a lipid? Well, other lipids. So cholesterol, other molecules like that can move simply through the cell membrane just by dissolving into the membrane itself. Another way that things can move across the cell membrane is via protein channels. These protein channels are especially important for things that are ions. Remember, ions are essentially atoms that have a charge because they've either given up or received extra electrons. And because they have a charge, they attract water towards them, and they won't be able to cross this hydrophobic bilayer right there, but they can go through these channels. And these channels are generally molecule or ion specific. So we might have one channel that allows potassium, one channel that allows sodium, one channel that allows chloride, etc. We even have special channels that allow water to pass, and these are called aquaporins. And basically they're inserted on the fly in the kidneys to allow water to move into or out of a tubule. Okay, another method for moving things across the cell membrane is a process called facilitated diffusion. Facilitated diffusion is very similar to the process we talked about before using the protein channels. The only difference is these protein channels undergo a shape or conformational change as they bind to the substrate on one side and spit it out on the other side. Now we see this happening particularly inside the kidneys where we have filtration of blood, and we tend to want to pull out the sugar molecules, the glucose, which are initially in the kidney filtrate, and put them back in the bloodstream. And so glucose will bind to these three-dimensional proteins. The protein will undergo a conformational change, and it will spit that glucose out on the other side of the membrane, putting it back into the bloodstream. Okay, just a review of plasma membrane function. We said before that the plasma membrane is a semi-permeable membrane which will allow some things to cross through but not others. And things can move through that membrane in various ways. One is through passive transport. Examples of passive transport were osmosis and diffusion. This is the downhill movement of solutes or water molecules going from an area of high concentration to an area of low concentration. So in order for this to work, we must have a concentration gradient, but the good side is we don't need any external energy. The second type we haven't talked about so far is active transport. This is the uphill transport of solutes or water or whatever, but it does require energy. So let's take a look at how that works. So imagine that we have 3 solute molecules on this side of the membrane, and let's say we have 8 over on this side of the membrane. By simple diffusion, things are going to go from high to low. But let's say we want to pull all of these sugar molecules inside of our membrane. We can't do that with simple osmosis or diffusion, but we can do it with active transport. So active transport doesn't require a concentration gradient, and it can push things from a low concentration to a high concentration, but it's going to require some energy to do that, and in most cases that energy is going to be ATP. So the primary example we have of active transport in the human body is something called the sodium-potassium pump. The sodium-potassium pump is a special protein which uses ATP to pump all the sodium from inside the cell to the outside of the cell, and all the potassium from outside the cell into the inside of the cell. In this way, we set up an ion gradient where we have mostly potassium in here and mostly sodium on the outside. We couldn't achieve this with simple diffusion because with diffusion, we would only get 50% of the sodium in here and 50% of the sodium in here. But using active transport and spinning ATP, we can create these large ion gradients and push all of a substance on one side of the membrane and another substance on the other side. Finally, endocytosis, exocytosis. I have this as a third form of transport. It is used to transport very large molecules from outside the cell into the inside of the cell. And think about amoebas and paramecia and stuff that have these huge food vacuoles that will literally engulf things. This also happens inside of white blood cells. So basically we form a vacuole of plasma membrane around whatever we want to consume, and then that pinches off and comes inside of the cell. So we would call that an endocytotic vesicle. On the other hand, we have something that we might want to export from the cell, but it's too big. Let's say it's a protein hormone or something like that. It would be inside one of these exocytotic vesicles. It would move towards the cell membrane, fuse with that membrane, and then be expelled on the outside of the membrane. So again, a lot of the proteins the cell manufactures are too large to move outside of the cell without using exocytosis. So think about the production of insulin, which is a protein hormone, made inside the cell, and it's going to be made within our rough endoplasmic reticulum, and then it's going to move via vesicles to the outside of the cell where it's expelled into the bloodstream. Okay, you've reached the end of the lecture on membrane transport processes. Before you go on to the next lecture, be sure you can define osmosis, diffusion, facilitated diffusion, and active transport, and be able to tell me what is going on during each of those processes. You should also be able to describe ion gradients as hypotonic, isotonic, or hypertonic, and describe what would happen to a cell if placed in each of these 3 solutions. Finally, you should be able to predict which way solutes or water will move when placed in a U-shaped container. Again, if we have more solute on one side, it's going to move from high to low, unless we're talking about something that's crossing a semi-permeable membrane. So make sure you review each of the slides in this lecture before you go on to the next.